Like any multicellular organism, plants have several different types of cells which are specialized in structure and function. And in plants, these cells are parenchyma, collenchyma, sclerenchyma, and then water-conducting cells of the xylem and sugar-conducting cells of the phloem. Parenchyma cells have a relatively thin and flexible primary cell wall and no secondary cell wall. So they're not as rigid as some other cells, and they're the least specialized. They often perform metabolic functions like photosynthesis, for example, and they often retain the ability to divide and differentiate into different kinds of cells. In Bio 131, you may have looked at parenchymal cells within Elodea, and you can see all the chloroplasts in these cells because, again, they're the ones that are doing the photosynthesis. Collenchyma cells are a little bit sturdier than parenchyma cells. They don't have a secondary cell wall, but their primary cell wall is thicker, although it can be a little bit uneven. And these are there to provide some support while also being a little bit flexible without restraining growth. And here you can see some collenchymal cells in the shoot of a helianthus. Sclerenchyma cells are the toughest, most rigid cells, and this is because they have a really thick secondary cell wall that has lignin to strengthen it. And lignin is a very, very tough type of carbohydrate. And these cells tend to be dead when they are functionally mature, and they can make up fibers. For instance, the hemp fibers that you use from rope are taken from these kinds of cells, or scleriids, which are little bits of cells with lignin around them, like you find in pears. That grainy stuff in pears are examples of scleriids. And here you can see an example of a scleriid from a pear under a light microscope, or fiber cells within an ash tree. And these are going to be structural kinds of cells that, again, are tough because of the lignin in the secondary cell wall and allow it to firm up the structure. In terms of water-conducting cells of the xylem, there are tracheids and vessel elements. Both are dead at functional maturity, and tracheids are found in all vascular plants. Vessel elements, on the other hand, are common to almost all angiosperms and some gymnosperms, but not the majority. And these vessel elements tend to align end to end, and they have perforations at end plates. And they form essentially these long micro pipes that we call vessels. Here you can see vessel elements and tracheids, and both are tubular and have pores in between the places where two cells come together. These tracheids tend to be longer and narrower and tapered at the end, whereas the vessel elements tend to be a little shorter and wider and not so tapered at the end. They just have these perforation plates with pits. But both of them are going to be dead at functional maturity. They're going to have thick secondary cell walls with lignin, kind of like your scleriids. So they're tough, and they allow a tube, essentially, for water to move up from the roots to the shoots. In the phloem, you have sugar-conducting cells, like sieve tube elements, that are alive at functional maturity. But they lack key organelles, so they need help to keep them alive. And so each sieve tube element has a companion cell, which is got enough organelles to essentially keep both cells alive. And they have these sieve plates, which are porous end walls where the two cells meet, that allow sugar water to flow from one cell to the next. So here you can see these sieve tube elements here with the sieve plate in between the two cells. And again, these are going to look a lot like your vessel elements in your xylem, except that, again, these are alive and they require companion cells to keep them alive next to them. Some plants can continue to grow throughout their lives. This is called indeterminate growth. Other plants grow to a particular size and then they stop, and that is determinate growth. And growth happens at meristems. These are places where a lot of mitosis occurs. They're embryonic tissues. Apical meristems are at the end. So you have a shoot apical meristem at the top and a root apical meristem at the bottom, and allows them to grow longer. And this growth in length is called primary growth, the growth of roots and shoots. Woody plants, like trees, can add thickness as well as length. And instead of apical meristems, lateral meristems are going to be embryonic tissue that is able to add width. And this adding of width, this thickening, is called secondary growth. And there are two main types of lateral meristems, a vascular cambium and a cork cambium. The vascular cambium adds new layers of vascular tissue, secondary phloem for sugars and secondary xylem for water. That secondary xylem is going to be what creates eventually wood. And cork cambium replaces the epidermis with the periderm, which is it's thicker and tougher, and that's going to be what gives us bark. So here you can see the shoot apical meristem at the top. The bottoms of these roots each have a root apical meristem. But then if you look inside the stem, you have the vascular cambium and the cork cambium. Vascular cambium is right here. It's producing a secondary phloem on the outside and a secondary xylem on the inside. And then the cork cambium is producing this periderm on the outside. Meristems are full of cells undergoing mitosis, and mitosis starts with a single parent cell and produces two daughter cells. So whenever you have that happen, one of those two cells is going to remain mitotic. It's going to stay as a stem cell so that you can keep doing this over and over again. And the other one is going to become specialized in nature. It's going to become a mature tissue cell somewhere. And so you have an initial which will divide, and one will remain an initial and the other will become a derivative. And then in woody plants, in addition to primary growth, you have at the same time occurring secondary growth. And this is why if you look at a tree and you're out there with your honey one day and you carve ill-advisedly into the trunk of a tree, M hearts M. My wife is Meredith and I am Mark. So we're out and we carve that into a tree, and we come back 20 years later, we revisit this romantic walk that we had a couple of decades earlier, and M hearts M is at the same height, right? It didn't go up as the tree grew up because that lengthening only occurs at the shoot and at the root. But the tree is much thicker than it was before when we initially carved our names in it. So here you can see growth from this year at the very top, growth from last year just below that, growth from the year before below that, from a couple of years ago. And again, this is going to stay essentially at the same height off the ground, and any new growth is going to occur up here. And then here, this is a branch that happened a year ago, right? And you sometimes lose old branches or leaves, and there are scars that remain there from that old growth. Some plants complete their entire life cycle in a year or sometimes just a season or two. And they grow, they flower, they reproduce, they create seeds or fruits, and they drop them, and then they die. And those seeds may grow the next year, but those are different plants. Sometimes you require more than 1 year for your full life cycle. Biennials require 2 growing seasons. You may have one growing season where you acquire a lot of resources, and in the second growing season you shoot up and you flower and fruit. Perennials can live for many, many years. So trees are examples of perennials. Bushes, a lot of them. Tomatoes are examples of annuals. If you zoom in to a meristem, like say the root meristem, the root meristem is going to have a root cap on the bottom of it. You don't have that in the apical meristem, but the root meristem has a cap to protect it as it digs down into the earth. But then you'll see just behind that a zone of cell division. This is where we're actually doing the mitosis. A zone of elongation where you grow not by adding more cells, but by those new cells getting bigger. And then a zone of differentiation or maturation where those new cells begin to differentiate in their structure and then their function. So here you can see a root, and you can see the dermal tissue out here and the vascular tissue and the ground tissue. And you can also see the root hairs that are expanding surface area. But at the bottom you can see this root cap, which is protecting this meristem. And then you can see this zone of cell division where mitosis is occurring. And you can see where these cells up here, which divided earlier, right, are elongating. This is the zone of elongation. And eventually those cells will differentiate. If you look at a cross-section of a root, you can see in dicots, it kind of looks like a little cross here, the vascular cylinder in the middle, whereas in a monocot, it's going to look a little bit different in its arrangement. Here, if you zoom into this vascular cylinder of a dicot, you can see this cross is made of xylem, and in between there are phloem cells. We also, in addition to this epidermis out here, have an endodermis that surrounds this vascular cylinder, and that endodermis is going to prevent certain things from coming into the xylem and the phloem. It's like certain stuff getting into your blood vessels or across your intestines. You don't want to allow everything through. You have to have some sort of scrutiny about what comes into your body and what doesn't. When you're producing lateral roots that come off of these existing roots, you have bursting from this pericycle and out of the endodermis, you can see this lateral root will begin forming its own tissues. If we go up to the shoot meristem, the shoot apical meristem, we can see these leaf primordia that sort of protect the shoot apical meristem. It kind of looks like devil horns a little bit. And you can see these axillary bud meristems as well. These are sort of waiting to grow. They're suppressed hormonally by the shoot apical meristem. But if this shoot should ever pop off, then these axillary buds will be able to shoot off. But here you can still see, in this meristem, you have this area of division and then elongation and differentiation, but you don't have a cap like you have in the roots. And while these leaf primordia, could become leaves, they aren't actually leaves. So the leaves that you see just around the bud are actually right here. These are young leaves. Now, if we look at a cross-section of a stem, you can also see ground tissue in the middle here, and you can see vascular bundles of vascular tissue around the outside. This is a dicot, and you can see on the inside is the phloem, or is it the xylem, sorry, and then the phloem, and these are both attached to some sclerenchyma, fiber cells. As we mentioned, these sclerenchyma cells have a lot of lignin in their secondary cell wall. They're very thick, and so this provides structure. And each of these is a vascular bundle, and you have a ring of vascular bundles with xylem, phloem, and sclerenchyma. In monocots, you still have vascular bundles, but the vascular bundles are not in a discrete ring. They're sort of scattered around the stem. Now let's look at a cross-section of a leaf to see the structure. So we have dermal cells on the outside. Again, these form the epidermis of a leaf and secrete a waxy cuticle on the outside to prevent this leaf from drying out. And you have these little pores called stoma, or stomata plural. That means mouth. And usually more of them are on the bottom to prevent too much water loss. And those are there to exchange gases. You're constantly in the leaves producing oxygen from photosynthesis, but you need to bring in CO2, so you have to exchange those so you don't end up with photorespiration, which you may or may not remember from Bio 131. And so you have stoma that you can open and close using these guard cells. Again, usually they're on the bottom of the leaf because this is an area where you can lose a lot of water. And on really sunny days, those guard cells might close up the stoma, so you're not exchanging so many gases. You also have these veins that are essentially full of xylem and phloem. And then you have mesophyll. And the palisade and spongy mesophyll are the stuff in between the dermal tissue and around the vascular tissue. And the palisade mesophyll is where most of the photosynthesis is going on. The spongy mesophyll has lots of gaps in it because it's where we have a lot of gas, again, that's coming in or moving out through the guard cells. That's why it's called spongy, and you can see what this looks like here. So all of this mesophyll is going to have a fair amount of chlorophyll in it, but especially the palisade mesophyll. It's going to be crowded up there with lots of photosynthesis going on.